Thanks, Annemieke. Okay, so today I'd like to tell you something about our efforts to apply proteomics in virology. And this is in fact only a side project, so please allow me to first, as a short introduction, give you a very quick overview of the activities of my lab outside of these hectic COVID times. So we are involved in a lot of interactomics or protein-protein interaction network studies, which usually serve as fishing expeditions and as the foundation for many projects. And something that is more and more common is proteotyping. So as we can cover a substantial part of the cellular proteome routinely now, proteotyping could be complementary or even serve as an alternative for genotyping. So for this, what we basically need to do is categorize and count as many elements, proteins in this case, as possible of a complex heterogeneous system as possible. And a branch of research that we have started a few years ago is Immunopeptidomics, where we analyze HLA peptides, for instance, in order to develop better vaccines. Quantitative proteomics, we do a lot, especially in combination with the analysis of post-translational modifications. So for any study to the dynamics of the flexible proteome, we need quantitation, and powerful methods to do so are SILEC and TMT and so on. Then the analysis of post-translational modifications is another main theme in the lab, which includes the development of enrichment tools for different modifications, such as phosphorylation, ubiquitination, methylation, with ubiquitomics being one of the main projects. And finally, targeted proteomics is something that we are really more and more interested in because of potential applications in translational and clinical research. So today, I'm going to tell you about 3 different approaches of mass spec-based proteomics in virology. I'll start with a short story about interaction proteomics that we did when we were studying MERS coronavirus several years ago. And then I'll switch to the main topic, which is the use of proteomics as a diagnostic tool for COVID-19. But first, let's go back to 2013. When in Jeddah, Saudi Arabia, a medical doctor was treating a patient who was coughing and suffering from high fever and who didn't seem to respond well to several treatments for known viruses at all. So they then decided to send some patient material to Erasmus MC Rotterdam, with whom they were running some collaborative projects already. And here at the virus science department, the samples were analyzed, and soon they found that they were dealing with a novel virus. And this virus was genotyped and later called MERS coronavirus, for Middle Eastern Respiratory Syndrome coronavirus. And it soon became clear that MERS was becoming another SARS-like threat to the world. Genetically related to SARS, it has killed over 900 people, which corresponds to a fatality rate of over 30%. However, the mechanism that was used by the virus to enter the human host cell was not known. And clearly the receptor protein for MERS turned out to be not the same one as SARS uses to enter cells. And this is in fact where we came in. So in order to identify its receptor on the human host cell outer membrane, we performed a interaction analysis. So the MERS coronavirus spike protein S1 is a type 1 membrane glycoprotein, and so we then used a rather standard bottom-up mass spec-based proteomics workflow to identify the unknown receptor. And this turned out to be a cell surface aminopeptidase, dipeptidyl peptidase 4, DPP4, also known as CD26. And so clearly different from the ACE2 receptor that is used by SARS and also SARS-CoV-2. This multifunctional DPP4, highly conserved among mammals, plays a major role in glucose metabolism by its degradation of incretins. And in humans, it's primarily expressed on the epithelial cells in, for instance, the lungs. Now, further in vitro and in vivo experiments that I won't describe here in detail confirmed that DPP4 indeed is the host cell receptor for MERS coronavirus. And this left panel over here shows that S1 binds to monkey COS cells, transfected with plasmids encoding DPP4, and then stained with an anti-DPP4 antibody. And the figure on the right here shows that DPP4 is essential for viral infection. And in green, cells that can only be infected when DPP4 plasmid is present in these bronchiolar epithelial cells. So the conclusion is obviously that the identification of MERS coronavirus receptor protein is necessary for the development of vaccines and antiviral drug treatment, for instance, by specific blocking of the receptor. But from a proteomics point of view, we can say that proteomics is more and more replacing more conventional tools because it is an unbiased tool. So if the virologists would have done screens for potential receptors, they would probably not have found it because they were not expecting it at all. And it took us all by surprise that DPP4 was actually the receptor here. Also, on the left here, we got our 50 minutes of fame because at that time, MERS was still called the coronavirus EMC, where EMC stands for Erasmus Medical Center. So this was actually the first time that the name of our institute was on a Nature cover. And only 3 months after this publication, the crystal structure of the receptor binding domain of MERS bound to DPP4 was published. Okay, so fast forward to 2020. Well, it's, I think, known to all of you that the causative agent for COVID-19 is this SARS-CoV-2 virus. And I guess you're all familiar with the numbers here on the right. Obviously, extensive testing for SARS-CoV-2 is crucial to define sites of outbreaks and to take measures to contain and stop the spread of the virus. So there was and still is a need for rapid, sensitive and specific diagnostic tests, and the golden standard of SARS-CoV-2 tests are PCR-based. Still, the coronavirus study group earlier in the previous year urged people to take a proactive approach to develop alternative and additional diagnostic tools. Not only because of insufficient testing capacity, but also for research purposes, of course. So as a proteomics lab, we asked ourselves whether mass spec could be used as a tool for testing. And in this talk, I will summarize what we've done so far. So the virus is built up of an RNA strand, several proteins, and EM. You've seen this in the previous talks already, surrounded by a lipid membrane. And the precursor protein ORF1ab is translated and then proteolytically cleaved into functional proteins. This cartoon is perhaps more realistic in that it shows the protein abundance ratios better. Nucleocapsid is by far the most abundant protein in this virus particle. And we confirmed this ourselves using label-free quantitation, and I will come back to this later. So we set off by defining the target tryptic peptides that have to serve later as the target peptides in targeted proteomics assays. And for this, we took Vero E6 cells infected with SARS-CoV-2. So the virus was first inactivated by boiling the cells for 20 minutes in a Lemley buffer containing a lot of SDS. And this would be our so-called test sample. The rest of the protocol is quite straightforward. Proteins are precipitated, digested into molar urea. And we were in fact very lucky that just before our institute was shut down last year, the brand new Orbitrap Eclipse was installed. And literally the first proteins that were measured on this beast were SARS-CoV-2 proteins. For most samples that I will describe later on, so samples consisting of difficult matrices, patient samples, we use the SP3 method that was developed in the Keisfeld lab, optimized for small sample amounts, and which works very well in our hands. This is just an overview of the results of the test sample. We could identify all SARS-CoV-2 proteins with decent coverage percentages. Apparently there's a lot of virus in these cell cultures, both in the cells and excreted from the cells. And I should emphasize here that we do not know how much of these proteins is really assembled in active or infectious virus particles. Based on these MaxQuant label-free quantitation or LFQ data, we think that almost 90% of the total virus proteome is composed of nucleocapsid. Now, for all of the following analysis that I'm going to describe now, we use targeted proteomics tools. So targeted detection of proteins or peptides of interest. And in PRM or parallel reaction monitoring on an Orbitrap, a peptide is of interest is isolated and the precursor is then fragmented. And fragment ions are collected and mass analyzed in the Orbitrap at very high mass accuracy and resolution. And this gives then a very specific and often unique fingerprint of the particular peptide shown in step 4 here. And because this fragmentation spectrum is so clean with no background contaminant peaks, accurate quantitation of the peptide can be done based on these fragment ion chromatograms on the right here. Now, is PRM a novel technique? No, not at all. In fact, it reminded me of this situation at home last week when I showed my 11-year-old son an old floppy disk. And he said, wow, cool, Dad, you 3D printed the save icon. So no, targeted detection is not new, of course, but what I want to stress here is that when using Orbitraps, both the selectivity and sensitivity have dramatically increased as compared to MRM studies on a triple quad. And this in fact opens up a lot of possibilities. So the difference, of course, being the fragment ions measured at high mass accuracy and resolution in the Orbitrap compared to the relatively low resolution in the third quad of a triple quad instrument. And the resulting peptide fingerprint in an Orbitrap is just much more informative than the fingerprint from a triple quad MRM assay. So illustrated again here in a blow-up of a Y fragment ion, in this case, in a peptide fragmentation spectrum. So these are just a few fragmentation spectra, and these MSMS fingerprints were used to design the PRM workflow and below the sequence coverage for nucleocapsid and VME1, another SARS-CoV-2 protein. So next we measured a dilution series of the cells plus virus test sample in a targeted PRM assay, where we focused on a few highly responsive nucleocapsid and spike peptides. And these are just two examples of those peptides here. For the lower limit, we could go down to about 20 picograms of total input protein amount. And this then would correspond to less than 1 picogram of nucleocapsid protein. These are the same results in a different format. You can nicely see the linear relationship between input and signal over several orders of magnitude in this PRM assay. And we then did some basic calculations to find out which number of viral particles this would correspond to. Don't pay attention to the exact numbers here. I make a few assumptions. So first, the composition of an RNA-based virus here. And then the assumption that about 90% of the SARS-CoV-2 proteome is nucleocapsid. So that would be over 1,000 copies per virus particle. And then the lower detection limit would be in this case about 10,000 virus particles in a sample. There are clearly a few big ifs here, and this should be considered no more than just a ballpark figure. Now, after these preparative experiments, we took this further to patient samples and asked ourselves whether we could measure viral proteins in them. And I must admit that this whole endeavor started off in a quite disappointing manner, as we weren't able to detect viral proteins in material from the first swabs at first. And the main problem was apparently that the diagnostic department delivers all patient material in UTM. That is a transport medium containing a lot of serum and albumin, according to their standard protocols. But for proteomics, of course, this increases the matrix, the background to a huge extent, and that's not what we want. So we went looking for other sample types and then received some sputum samples. And we had already seen in the literature that sputum or saliva are maybe even better sources of material for virus detection. So for these samples, the patient basically just spits on a glass slide, and the material is then fixed in 80% acetone. And the first results actually looked quite promising. So we targeted several nucleocapsid and spike peptides. And this here is just one of those target peptides as an example. So using PRM, we could nicely detect the target in the glass slide samples. And what you have to look at here is the fragment ion chromatogram pattern and the retention time that should correspond to the positive control sample. So you can see that the fragmentation pattern in the patient sample is virtually identical to that of the positive control sample. If you compare this to the results of the swab samples, it is clear that sputum on a glass slide as a sample works much better. So these are sputum samples here, in which we could all detect the peptide, and these are swab samples in which we could not. So we then incorporated the use of heavy stable isotopes as spike-ins in the sample for two reasons. So that it makes it possible to more accurately identify and also quantify target peptides. And this is what we did for, in this case, the same target peptides as I showed you before. So these are the chromatograms of the heavy peptides that you see over here. And these are the target peptides, the endogenous peptides, with clear signals for the sputum samples and less to no signals for the swab samples. And I'll just switch back and forth a few times so that you can compare the plots. So these are the endogenous peptides, and these are the spiked in heavy peptides. So the patterns are very much identical here. Next, we analyzed a batch of e-swabs kindly provided by the Jeroen Hospital. And the huge advantage of this kind of swab is that there's much less background contaminating proteins in them. So we tested swabs with various e-gen CT values using the same PRM assay for several target peptides. And the result for tryptic peptide GFY here of nucleocapsid is given in the left panel here. So the upper panel displays the aqua peptide intensity, so the spike in, and the lower panel, the endogenous peptide signals for the 11 patient samples that we measured. And these are on the right here, several other peptides showing the same picture, huge signal in samples 5, 1, 5, 10, 11, and virtually no signal in some of the others. Since some fragment ion signals are really low, we check all of the fragment ion chromatograms in the Skyline output manually as well. by comparing the patterns of the aqua peptides with those of the endogenous peptides. And both the relative intensities and the retention times should fit. If you then sum the intensities for all peptides and plot these against the corresponding e-gen CT values of the same samples, then these are the results. So obviously for the higher CT samples, so that means more virus present, the intensities are high. And for some high CT values, less virus present, the intensities are low, or we couldn't detect any peptides at all. Some of them are plotted here based on the automated Skyline output, but manual inspection is more stringent. Then for a second sample cohort of E-swaps and Optima swaps, another type of swaps, these were kindly provided by the STAR SHL diagnostic labs in Rotterdam. We basically performed a similar analysis, and the results are given here. As you can see here in these zoom-ins, for several samples of CT values higher than 20, we could reliably detect and quantify peptides. Now let's take a closer look at a few samples that contain relatively low amounts of virus, for which we carefully checked all spectra. So for instance, sample number 7 here. If you take number 2 as the positive control, because there's so much virus in there, these are the chromatograms for 2 nucleocapsid peptides. And sample 7 contains much less virus, and the intensities in the chromatograms are much lower. But still, I hope you can appreciate that the same pattern here is visible in sample 7 as in sample 2. And even for this peptide here on the right, if you zoom in, I can't see my screen, but I hope you can see it. If you zoom in, then and discard the blue background peak, then the same pattern is visible. Now another example is number 4 with a CT value of 29, so very high, not much virus present. For this peptide, we spiked in the aqua heavy counterpart peptide, of which the pattern is given here. And note that the Y4 and Y6 ions are on top of each other because they have virtually the same intensity. Now for the endogenous peptide, Skyline gives a rather large mass error. However, the blue chromatogram seems to be a contaminant, and if you discard that one, you can see that the other 4 fragments with very small mass errors, shown here on the right, show the right pattern, which makes us conclude that this indeed is a positive hit as well. So we have also performed shorter LC-MS gradients to decrease the total analysis times. And this gave more or less the same hits, although some peptides were missed. This is again number 4. And you can see here that there's obviously more co-eluting peptides present there. Finally, we are trying to increase the sensitivity of detection by offline peptide fractionation of the samples. And as you can see here, using fractionation, we decrease the sample complexity. And in this case, we can detect the peptide in a relatively low viral count sample that remained undetected in the non-fractionated sample. The same is true for another peptide, this EIT peptide over here. And with the use of aqua peptides, we can of course also do absolute quantitation of the peptide. And this amount of peptide, which is really at the boundary of detection, would then correspond to roughly the low attomolar range. And this then corresponds to the limit of detection that we observed in the dilution series that I showed you earlier on. This is a peptide from a higher viral count sample, and from the peak areas we conclude that using fractionation we may gain another 2 to 4 times in sensitivity. So there's still work to do. So in conclusion, yes, we can detect viral material in patient samples. And well, that's great, of course. And even at relatively high CT values, so low viral counts, we often can detect at least a few peptides. Still, in order to be successful as a diagnostic tool, we need to make several improvements in terms of increasing the detection limits, reducing the sample complexity, and also the analysis times should be decreased. And finally, we are currently trying to find out which sample types show the best results. In principle, any sample type, anything goes, as long as we can extract protein from it, of course. What I think would be the most interesting application, though, is what could make this technology unique as compared to conventional testing in this field, is the screening for multiple viruses simultaneously. So, for instance, to be able to quickly determine which virus is causing an infection. Besides SARS-CoV-2 peptides, target peptides, you could easily target influenza, MERS, any peptide you can think of. This is the very last slide that shows another contribution of proteomics in SARS-CoV-2 research, which is in this case a study of the host cell response upon viral infection. And I'm not going to talk about this in detail, but what we did here was a proteomics analysis of an organoid-derived bronchio-alveolar tissue culture infected with virus to study the induction of the interferon response program. With this, I'd like to end. Most of the mass spec work was done by Karel Besterosti in my lab. Much of the virus work by Mart Lames in the lab of Bart Haagmans. And many of the patient samples were kindly provided by Drs. Peter Wever and Hua Tai. Part of the work was posted on bioRxiv some time ago, and the manuscript is on the submission now. Thanks for your attention, and I'd be happy to take questions.